Hello, my name is Yong Mi Kim. I'm a professor of pediatrics and pathology at Children's Hospital Los Angeles, University of Southern California. It's a pleasure to be here and to share the highlights of the research of my lab. Drug resistance in acute lymphoid and myeloid leukemia cells remains a problem. Interestingly, most of the relapses occur in the bone marrow. There's something very particular about the bone marrow that acts as a shelter for leukemia cells and lets them withstand chemotherapy, which is why my lab focuses on the bone marrow as a safe haven for leukemia cells. In particular, we have been studying the interaction of stromal cells with leukemia. We have developed several models that help us study drug resistance. Here we show patient-derived leukemia cells in green after a week of chemotherapy treatment. The contact to stromal cells shown in red allows some leukemia cells to resist chemotherapy and survive. So our hypothesis is that leukemia cell-stromal cell interaction mediates drug resistance and that dislodging leukemia cells from the chemoprotective haven could make them vulnerable again to chemotherapy. The question is, what are the major anchor molecules that let leukemia cells stick to the bone marrow cells, and how can we target them? To answer this question, we focused on a major adhesion protein family called integrins, and the role in cell adhesion-mediated drug resistance. This figure shows you an example of our research when we treat leukemia cells not only with chemotherapy, but with an antibody against an anchor molecule in integrin alpha-6. We can actually prolong the life of the mice significantly and almost cure them. This is very supportive of our hypothesis that the microenvironment contributes to the drug resistance of leukemia. So far, we have looked at integrin alpha-4, integrin alpha-6, and their role in drug resistance of leukemia. We have studied the downstream signaling of integrins and also branched out to the role of integrins in immunotherapy and in biophysics. These are very exciting areas, and I'm very happy to meet with collaborators to develop our preclinical evaluation and to develop our targets further for clinical care. We have a poster here, and I'm happy to answer your questions.